Hello, people. I'm not expecting anyone. Not expecting anyone today. I'm going to post this later today. I hope you're all enjoying the apocalypse. Oh, here's David Roy. David never misses. David, as I was just saying, you're not expected to be here synchronously because of the apocalypse, but I'm going to be posting this later. Have any questions? Remember, David, I don't see the chat, so if you've got a question, go ahead and shout it out. More than happy to slow down and start over. Okay. Let's kick it off by talking about last weekend's quiz. Bum, bum, bum. All right, I'm sharing my screen. Let's go to UVM. I should have had this up, sorry. Exam 4, quiz 1. Preview. Okay. Hope I know the answers. It's been a while since I looked at this. Which phospholipid is read as an eat me signal for phagocytes? So we talked about this way back in chapter 2. I love themes that keep coming back again and again. I think it makes it easier to understand things when you see the whole process rather than just little factoids. Okay, what is the role of SMAC Diablo? SMAC Diablo is going to inhibit the inhibitor of apoptosis proteins. And remember, SMAC Diablo is stored in the mitochondrion and it gets released the same way that cytochrome C gets released. So activating the apoptosome as well as inhibiting caspase inhibitors. If a metastatic cell from the skin is taken up by the blood, where is the likely site for it to set up new tumors? So we said liver, because of that hepatic portal vein, anything from the intestines is going to go to the liver first. That's a good place to set up a new tumor. Lymph nodes, and which is not, which is a likely site for it to set up. Wow, all of them except bones, I would say. Bones happens with some, but not all. Wow, this is a bad question. I think it should be which is not a likely site. I'm going to say lungs, but I'm going to have to go through and give everyone credit for that one because that's a bad question. Okay, which of these is the most dangerous mutated cell? That would be neoplastic. Neoplastic. It goes hyper, meta, dis, neo. Oops, too far. These domains are found on caspases that are activated by the extrinsic pathway, extrinsic death domain. So there's the death domain and then there's the death E domain. E, I always forget what the E stands for. Hello? Okay, PED. Which phospholipid has one fatty acid cleaved off by PLA2 and is then released as a find me signal? That would be the choline. Lysophosphatidic choline is released by cleaving off that middle fatty acid of phosphatidylcholine. Which phospholipid is in the autophagy program and ATG is bound to it? And that is ethanolamine. So chapter 2 coming into play here in this chapter. Which domain on the BCL2 protein is primarily responsible for the homodimerization? That would be the third one, BH3. Oh, another phospholipid question. Which phospholipid is phosphorylated in the autophagy program to attract WPI and the E3 ligases? Phosphatidylinositol is going to be phosphorylated on that 3 position. Okay, which of the following is activated by caspase 3? This is going to be broken down. This is going to be broken down. This is going to be activated. ICAD. No, this ICAD is inhibited. So ICAD is the inhibitor of caspase activated DNase. So that's active until caspase 3 inhibits it. So gelsolin is activated, and that causes breakdown of the cytoskeleton. All right, let's see how I did. I think that one question was probably wrong. Okay. How did I do, though? Okay, here we go. Oh, I was right with lungs. Although, again, liver and lymph nodes are good answers here, too. It was supposed to be not a likely site. So I will give everyone credit for that question.
Dr. Slish.
Yes.
Hey, good morning. Sorry, I missed your explanation on the metastasis question. Could you please
Sure.
re-explain what you had shared?
Absolutely. Let's start this over again. I did start a little quickly today. I wasn't expecting people to show up. So I'm like,
It's my work from home day, so I get to actually attend class.
Nice. Okay. All right. Preview.
Yeah, I was conflicted. I kept like changing my answer back and forth between lymph node and lungs.
Oh, okay. Yeah, that's a bad question. That's why you were struggling, because 3 of those answers are correct and one's wrong. It should have said which, let's just do this again. Pick it up. Question 3, I believe. No, it changes the order. This one. Oh, from the skin, taken up from the skin. Oh. Maybe this wasn't such a bad question. From the skin, it tends to go to the lungs. Okay. If it's the intestines, it would go probably to the liver. Why not lymph nodes, though? Still, I'm not crazy about this question. Metastatic taken up by the blood, where is it likely to go? It could go to the lymph nodes as well, I believe.
Sorry, go ahead.
No, you go ahead.
Oh, because I was like thinking, like, if you have like excess interstitial fluid, that interstitial fluid would get reabsorbed by the lymph nodes, right?
That's
Which
right.
does have some plasma in it. So
That's right.
yeah, that's
Yeah, lymph nodes could be a good answer here too. Yeah. I think bones is just a distractor. Liver is true if it's intestinal, but I think for epithelial, it could be lungs or lymph nodes. So I think what I'm going to go ahead.
I was gonna ask, is the lymph nodes more of a way of its transportation, or would they actually set up tumors like actively in the lymph nodes?
I think they actually set up tumors there as well. Although they could go through the lymph nodes and then get dumped back into the circulation. But I think in general, that's like my way of thinking is the first place they get slowed down. Because if you figure they go into a lymph vessels get bigger and bigger and bigger until they get to the lymph node. And that's the first place things get slowed down. Same thing with the blood. It gets into the veins, they get bigger and bigger, goes to the heart, then it goes to the lungs. And that's the first time you get to another capillary bed.
Thank you.
But I'll go through and give everyone credit for either of these because I think they're both good answers.
That's
okay. Any other questions about this quiz?
Yes, I have.
Go ahead.
Can you not hear me?
I can hear you. Yeah, go ahead.
Okay. So I was just wondering, the caspase 3 one, was it the gelsolin or ICAD? Though that was the one that was
a little tricky too, but it is definitely gelsolin, and I'll tell you why when I find it. Have I gone? Here it is. So it's gelsolin, and the reason is ICAD also gets cleaved by caspase 3, but that inhibits it. It doesn't activate it.
Oh, okay.
Yeah, and then also AKT and PARP are going to get cleaved by caspase 3. So all of these are cleaved and inactivated. This is the only one that's going to be activated. Okay.
Okay. Thank you.
Sure. Any other questions about the quiz? Okay, let's move on then. Questions in general? Any questions, or you want to do a quick review of the cancer that we did last time, and then we'll move on. And I hope we don't finish this chapter today, because I don't want to go too fast. But you know, whatever. Okay, so we started talking about the different classifications of cells as they move more and more towards cancer. So the most broad classifications then are benign versus malignant. And I keep harping on this basement membrane. This is the key. Carcinoma in general is a cancer of the epithelial cells, and that basement membrane is separating epithelial cells from the inside of the body. So at least in terms of carcinomas, once it crosses that layer, that's when it's really dangerous, because now it's on the move and it's invading tissues and potentially going the next step, which is metastasis. So then we go into the different types of changes in cells, I guess you would say. Hyperplasia, the cells are just dividing too much. Here's a normal milk duct in the breast, and here is a hyperplasia where the cells are dividing and going into the lumen of the milk duct. Your basement membrane is on the outside of this duct. So they're dividing, but they're going inside and they're blocking the duct there. Not dangerous necessarily, still benign, but on its way to becoming more dangerous.
Quickly,
Next is, I'm sorry?
are those treated in terms of like what gets done with them? Like,
honestly, when it comes to the clinical aspects, I'm not really up on that. So I really can't answer that, sorry. Again, you know, whenever you ask the question, I'm trying to come up with whatever I know to help you. One type of breast cancer is called ductal cancer in situ, and we'll see cancer in situ later, which is when they are very cancerous, but they're contained within that basement membrane. But yeah, I don't really know that much about treatment, sorry. Okay. So then metaplasia is we're not crossing the basement membrane, but we are crossing across borders, and cells are going where they shouldn't go. Here's an example, which is Barrett's esophagus. That's when esophageal cells are going across that cardiac sphincter and into the esophagus. And that can lead to esophageal cancer, which is deadly. It's not a curable thing. Okay. That is metaplastic. Dysplastic, now the cells are definitely wrong. These are abnormal cells. You can see abnormal nuclei, which tells you that the DNA is probably messed up. And you can see they're growing down in. Now that basement membrane is still intact, but they're pushing their way into the tissue. Or they could push their way out, which is a papilloma. So again, these are just cells that are dividing too much. They're screwed up, weird cells. You know, normally our epithelial cells are round at the bottom and they flatten as they go towards the surface, and these are just staying round. Here's your papilloma. Okay, so here's neoplastic in situ, carcinoma in situ. So these cells, basement membrane still intact, but these cells are definitely beyond dysplasia, and they're neoplasia. So this is like that ductal carcinoma in situ. The cells are definitely carcinogenic, and they're just looking for a way to get through that basement membrane. And then here is your malignant malignant invasion. This is neoplastic. And so they've come through. These are cancer cells. And you can see in this slice, you've got cancer cells inside the stromal tissue below it here. That's invasion. And then, of course, our metastasis, which involves crossing several barriers. We'll talk about crossing barriers today, if we get that far. And then I said, you know, they're usually going to end up in the capillaries or the lymph nodes. So I have to change the answer to that question later. Okay.
I have another question.
Okay.
So in the metastasis, the cancer is traveling to the bloodstream. When that's happening, do the cancer cells have the same morphology or the properties as the red blood cells?
Oh, definitely not the same as red blood cells. No, they're still going to be the same kind of screwed up cells that they were in the tumor. And there'll be, large nuclei, probably maybe even 2 nuclei. They're going to be really weird cells.
Okay.
So they're not changing.
And our immune system can still attack them, right?
Yeah, although typically by the time they've reached this phase, they found ways to get around the immune system.
Okay.
That's another one of the mutations that happens that helps them to, become cancerous.
Okay. I have another question as well.
Okay, just 1 second. So, for example, they may mutate out the tumor, the protein FADD, right? That would be a tumor suppressor. So, if that gets mutated out, then immune cells can no longer activate that extrinsic pathway, and they will have escaped it. So that's just an example. So you have another question?
Yes. So I couldn't attend the lecture last time, so I was listening to the lecture, and then when the cancer cells are innervating, are they like, is blood vessel or like bloodstream work like a chemoattractant to them that causes them to go into the bloodstream, or they just happen to come across like blood vessel or bloodstream based on like their innervation just by like sheer luck?
That's a really good question. I have no idea. I've never heard that question asked before. That's a really good question. What makes them go into the bloodstream? Yeah, I don't know. Yeah, good question. All right, let's call it a day because now I'm sad that I don't know the answers to all these questions. No, I'm just kidding. One of the keys to becoming an adult is not feeling bad when you don't know the answer to something. I don't know it. I know other stuff. Okay. Any other questions here? Because now we're going to go into the new material, something starting today. Okay. So the next step, then, we're going to talk about what changes occur to cancer cells, to cells that make them more cancerous and leading towards metastasis. So what are the changes in cells? One thing to keep in mind is the difference or tissue differentiation. So we'll talk about this again in the next section that I'm going to do. But differentiation, I just have this, let me blow this up a bit. This figure shows the differentiation of cells from mesenchymal stem cells, which are kind of in the embryo, through mature cells. And what differentiation has to do with is different hormonal influences on a cell, right? This mesenchymal stem cell is pluripotent, which means it can become a lot of different things. What it becomes depends on kind of the history of hormonal interactions it has. So not just one hormone, but a whole stream of hormonal interventions. And each of the hormonal activations is going to turn on a different set of transcription factors. And then the next hormone will then play off that change in transcription factors and use some of them and turn on other ones. So it's all about the history of that cell that determines its differentiation. So just for example here, this generic cell, if it's hit with FGF and TGF beta, becomes a myogenic precursor. At this stage, if it's hit with BMP7, it heads down the pathway to become brown adipose. If it isn't hit with BMP7 and it stays in FGF, it becomes a myoblast. And then depending on what it sees after that, it can become different types of muscle cells. So differentiation involves going from this early generic cell that doesn't really do anything in terms of maintaining homeostasis in the cell, right? It's not in the body. It's not contributing to the physiology of the body and keeping the cell alive. It's keeping the body alive. It's just there so we can make more cells, and the other cells are then the ones that are important for survival. So what happens then in cancer is what we call de-differentiation, where cells go, instead of going to this and down and becoming adult and stopping cell division, they stay back into this range over here where they're not functional, and they keep dividing. So the example I always like to use is white blood cells, right? You might think if someone had blood totally full of white blood cells, they'd have a kick-ass immune system, but they're immature white blood cells. So they don't perform a function, they just fill up the blood. And that's where they get the name leukemia. You've got white blood because it's so full of cells. But anyway, here is normal differentiation of epithelial cells. You've got your basal lamina here and your stem cell layer right on top of that basal lamina. And every time one of these cells divides, one goes up and the other stays here to be a stem cell still. And the one that goes up starts to differentiate. The shape of the cell changes, it flattens, it fills with keratin, and eventually sheds off the surface. But what happens in cancer then is the cells divide and they don't differentiate. They stay in this kind of undifferentiated state, and they just keep dividing and dividing and dividing. So that's one change in cancer cells that happens, is they stay kind of undifferentiated or more embryonic in character. Another thing that happens is two things about cells in tissues. One is they're always connected to extracellular matrix through their focal adhesions. I keep harping on this throughout this class because focal adhesions are so important for so many things. One of the things the focal adhesions are doing, as I've said before, is activating signal transduction. And that represents the trophic factor we talked about when we were talking about apoptosis. Activating, by being attached to extracellular matrix, they're activating these kinases, SRC and focal adhesion kinase, and they activate all of these pathways we've talked about before, and that's the survival signal. It's like, all right, we're connected, we're safe, we're good. Another signal that happens is between cadherins. So cadherins touch each other, and it's cell-to-cell interaction, and that tells the cell, all right, stop growing now. You know, the tissue has enough cells, don't divide anymore, we're done, we're good. So you got both of those signals going on. One is anchorage-dependent growth, and the other is density-dependent inhibition. And what happens with cancer cells is they lose both of these. So in terms of anchorage-independent growth, normally if a cell was taken out of a tissue, so it wasn't touching any collagen anymore, it would go into apoptosis. You would lose all of this trophic stuff going on here. And then remember how AKT and MAP kinase are phosphorylating bad, which is your pro-apoptotic BCL2. All of that shuts down, bad builds up, cell goes into apoptosis. And also, so basically that means that once the cell loses this, it can let go of its, once it has self-sufficient growth signals, this stuff is all turned on in the absence of connecting to collagen, then the cell can let go of the tumor and then crawl around in the body, in the cell, and become metastatic. So that's an important aspect of carcinogenesis, which is anchorage-independent growth. The other thing is loss of density inhibition. So rather than stopping growing when you're all packed together, they keep dividing anyway. And when that's what causes a tumor, right? Like, so you start forming a bulge where that tumor is because cells are building up where they shouldn't be. So that's loss of density dependent inhibition. So both of those have to do with signal changes in signal transduction that allow the cell to survive and keep dividing against these former signals. Here's an interesting side thing, which I don't know what it means. I don't think it means anything, but it's kind of interesting. Beta-catenin. Remember beta-catenin, our favorite transcription factor? Let's see, is it over here? It is not. It is here, but you don't see it. But beta-catenin typically is a transcription factor that activates signal transduction. Here it's a structural protein, which is kind of weird, but it's got two different roles in the cell. I don't know if those roles are related in any way, but just kind of an interesting little factoid there. Okay, loss of density-dependent inhibition. The other thing is telomerase. This is one that's gotten a lot of attention, or at least got a lot of attention 15, 20 years ago. And basically it has to do with preserving the cell, helping it to remain immortal, basically. The problem with the eukaryotic genome is that it's linear. Yeah, the prokaryotic genome, it's a circle, and you start at one end of the circle. After you keep going straight, you end up in the same place you left off, right? That's what a circle is. Here with our linear chromosomes, you start at one end. Well, you've got your leading strand and your lagging strand. So as this replication fork moves in this direction, the leading strand goes straight through, goes right off the end. But in the lagging part, you're going to end up with a gap at one end because your DNA polymerase can't start right on the end. It needs a primer to start from. You need that free 3' end to start adding bases to it. So in normal replication, you've got an RNA polymerase there that's going to lay a short stretch of RNA, and that RNA then is the end that DNA starts to go from. So you got that RNA free 3' end, and then the DNA completes the DNA strand from there. But enzymes go back and take that RNA out. So if you are replicating, every time you've got this RNA primer at the 5' end, it gets taken out, and you end up with a gap at that 5' end. So it's a little bit shorter. So every round of replication, your chromosomes get shorter and shorter at the ends. And this is something that they figured out in the 50s. You know, if they got the first step to figuring out how to culture cells that weren't cancer cells was you have to give them growth factors. Okay, that was step one. Once they figured that out, they realized that these cells will divide about 50 times and then they die. Okay. And the reason for that, they figured out later, was the chromosomes keep getting shorter and shorter and shorter. So how is it we have cells if we die after 50 divisions? Well, we've got an enzyme that's going to help protect those 5' ends, and that's called telomerase. So telomerase is going to be activated, or its transcription will be activated during S phase. So as you're replicating, this is getting turned on as well. So it's related to growth factor stimulation of the cell. So anyway, here's your telomerase. It's got an internal RNA template in it. And if you look, it's got ATC, which base pairs with TTG. And as the reverse transcription goes, it makes this, it uses the template to add more DNA. And then the last 3 bases it puts in are the same as the last 3 from where it started. So it starts out TTG, GG, TTG. So now this enzyme can come down and base pair with that template again, and put some more bases down, and go forward and put bases down, go forward and put bases down. So it does that, just kind of leapfrogging down the template, adding not random, but tandem repeats is what it's called. The sequence is repeated over and over and over again at the end of the linear chromosome. And so once you do that, then you can put an RNA primer here and then do your DNA polymerase like you did before, take out that RNA primer, and you've extended that end. And also there's this other little mechanism where it loops back around to protect the end as well, because you've got that repeated structure that allows it to base pair with itself. So basically this telomerase is adding these telomeres, or these, I think it's 6 or 8 base pair tandem repeats on the end. Now here's a little history for you. This was one of the first animations I made, and honestly, I don't even have this anymore. I have to go to this Wayback Machine to find it. Years ago, our server at Plattsburgh State changed, and I lost a whole bunch of files. So the Wayback Machine is an archive where you can go and ask for any particular website that existed since the World Wide Web started, and you can re-find it that way. So let's watch this. This is my animation. Let's see if I can get there. Wayback Machine, nope. So where did I go? So so this is a GIF animation, the classic old GIF or GIF animation, where it's like I drew a picture, saved it, moved something a little bit, saved it, moved something a little bit, saved it, moved it, saved it, moved it, saved it, moved it, saved it. Extraordinarily tedious. But I know I'm bragging, I'm sorry, but I get a kick out of this thing. All right, so here we go. Let's blow it up a little bit. So here you can see, and actually I messed up you can tell I messed up the template because there's a T, that should be a U there. So it leaves base pairs that it can re-anneal to. And it skips on down the line like that. There's your tandem repeats. Now we can put down a new RNA primer and protect those ends of the chromosomes. Okay. Ta-da! So point being here that another thing these cancer cells have to do is activate telomerase. Otherwise, they're going to die after a certain number of replications. And once again, that's going to be connected to growth factor activation or growth pathways activations. Blah, blah, blah. And that's what I'm talking about next. So the other thing that cancer cells do is they will get self-sufficient growth signals. So normally a cell has to be signaled from externally to be divided. These are kind of short-circuiting that. And what does that mean? That basically means any protein along this pathway could be mutated in such a way that makes it constitutively active, so you can never turn it off. And if that happens, then everything downstream happens to it, even if you don't have growth factor out here. You could also start way back at the growth factor. There are cells that will secrete growth factor and stimulate autocoid division, autocoid cell division, just based on releasing the growth factor. So one thing that's going to happen is you're going to have conversion of, so proto-oncogenes, let me define that. So a proto-oncogene is, well, an oncogene is a gene that produces a protein that causes the cell to become cancerous. And oncogenes were found first. Sarc was the first oncogene found. It was in a virus, the Rous sarcoma virus. But, you know, they found all these oncogenes in the like 40s and their 50s, and they're like, oh my God. And then eventually, when they looked at the human genome, they were like, oh my God, our genome is filled with oncogenes. How is this possible? And then slowly we came to realize that all of that, we've got these proto-oncogenes in our genome that can be mutated and turned into oncogenes. And these proto-oncogenes are basically any protein along this positive growth regulation pathway that gets mutated and can no longer be regulated. So that's one thing that's going to happen, conversion of proto-oncogenes into oncogenes. And again, any protein along this pathway could be that, and we'll see more later. And that, when you turn that on, right, you're going to inhibit apoptosis also. From our last, from our apoptosis lectures, you know that, you know, MAP kinase inhibits bad, AKT inhibits bad. We're always inhibiting apoptosis when these pathways are on. The other thing is the loss of tumor suppressors. So tumor suppressors, of course, are blocking cell division. When you lose them, you lose control of that process. So conversion of proto-oncogenes to oncogenes, loss of tumor suppressors. And sustained angiogenesis. This is also extremely important. So a tumor can only get to be about 1 to 2 millimeters in diameter before it needs to get a blood supply. And that's because of the diffusion of oxygen through tissues. Oxygen can go about 1 or 2 millimeters before it starts to run out. And so typically these small tumors will have necrosis in the center, where the cells, you know, the cells just keep dividing because they can't stop themselves, but the cells on the inside don't get enough oxygen, so they die. So one of the things that hypoxia does, though, is it causes the, it stimulates the release of angiogenic factors. Let's see if I have them listed down here. VEGF is one. Let's see, I don't have them listed here, but there's endothelial, trying to remember. Well, let's see if I have it in the next one. I know VEGF is one. Oh, I don't have them here. I'll have to list them later. But there are pro-growth regulators and anti-growth regulators. The pro-growth regulators are VEGF, vascular endothelial growth factor, and I think FGF perhaps. I'm not sure. There's another one. I'll list it later. And then you've got anti-growth regulators like angiostatin and endostatin. These are hormones released by cells that stop the growth of endothelial cells. But a tumor will start releasing like VEGF, for example, and that will cause this endothelial cells to start to divide and to crawl towards the tumor, right? Chemotaxis, and then they'll go into the tumor, form capillaries, and then they become just another part of the circulatory system. And at that point, the tumor can get as large as it wants, and it's got an access to a blood vessel, so it can metastasize out of there once it starts to cross that basement membrane. Here's a really great class of cancer drugs. I know personally a woman on Avastin. Basically, these are drugs that are meant to block that angiogenesis signal. And once the tumor loses its ability to, attract blood vessels, it starts to die. So these are really awesome drugs. Avastin is one. Bevacizumab. Here's an interesting tidbit too for you if you become doctors. If it says MAB on the end, that means it's a monoclonal antibody, so it's a protein. If it says IB on the end or US, it's a small molecule. So your proteins are going to have to be infused by IV versus the drugs. I'm not sure if you can take them orally or not, but I think a lot of them are oral, like inhibitors of these kinases are orally available. But anyway, that's not important for this class. I'm just saying the important thing of cancer therapy then is to block this angiogenesis. Okay, so that's, let me go over that real quick. Changes in cancer cells. Oh boy, oh boy. Changes in cancer cells, they become dedifferentiated. They lose their anchorage dependence. They lose their density inhibition. They activate telomerase so they can become immortal. They stimulate blood vessel growth into themselves. Oh, and they, oops. And they have self-sufficient growth signals. So those are changes in cancer cells. Okay, so now for invasion and metastasis, three things have to happen. They have to let go of their connections, they have to start crawling, and they have to clear a path in front of them. So first of all, let go. When we were talking about these, again, I'm sorry I keep harping on focal adhesions and stuff, but they're just so interesting. So your focal adhesion, there's actually communication going in both directions. In the direction we keep talking about, the fact that it's connected to extracellular matrix gives it the signal that tells it to survive. But also, the cell can tell the focal adhesion to let go, right? We saw that in cell crawling. At the trailing end of a crawling cell, it lets go. And it's not just snapping off those integrins. The integrins are letting go, and they're getting transported back to the front of the cell. And then also our cadherins are connecting cell to cell. These will also let go. So the cell lets go of its fellow cells. And then it's going to increase its motility. Getting back to chapter 3, Rackrose CDC42. So activating those signal transduction pathways often activates these three G proteins, and these are going to convert the normal cytoskeleton into a crawling cytoskeleton with our lamellipodium, stress fibers, and filopodia. So that's increasing our motility. And here's our cell crawling again with our focal adhesions. We protrude out with the lamellipodium, connect with the new focal adhesion, and then pull the cell forward into that lamellipodium, letting go behind. Okay, and now the other thing is you need to clear a path. So, you know, we have to keep in mind that our cells are not normally able to move around anywhere. Every cell is surrounded by extracellular matrix proteins that are holding it in place. And they need to be able to digest that or break that down to clear a path in front of them. The way they do that then is by releasing plasmin or tissue plasminogen activator. So we need to get through this. We need to break down the extracellular matrix, but also break down the basal lamina to allow the cell to get through that. Oops, too far. Way too far. Okay, here we are. So how do we do that? Well, there's these proteins called matrix metalloproteinases. These are kind of, what do you call them? Zymogens. They're zymogens. They're inactive proteases that are attached to the extracellular matrix. And tissue plasminogen activator activates plasminogen into plasmin. Plasmin then activates the matrix metalloproteinases, and that breaks down the collagen. So the tumor cells are going to release TPA, and that's going to activate plasmin, and then it can clear a path so it can crawl. TPA, by the way, is another drug. I'm pretty sure it was like the first genetically engineered drug that was released on the market, and it's used for to break down clots in, like a heart attack or something like that. You can give them TPA, and that'll activate the plasmin and break down the clot. So that's another drug. I know, pharmacologist, you always have to bring up drugs. So anyway.
I have a question.
So anyway, so the three things we need to do then are three things we need to do: let go, crawl, and clear a path. Okay, that's the next
Professor
one.
Slurs?
Yes.
I have two questions. So the first one is, you talked about how in cancer the coherence, like connection between the signal transduction is severed, and then you also mentioned like the cell needs to crawl.
Right.
What allows them to selectively like when crawling they also need to attach with something?
Right.
So what allows them to selectively like sever the connection the first time and then allow the connection next time when they are crawling?
Right. That all has to do with your RAC-RO and CDC42 activation of that whole pathway. Because you can see in the normal crawling sequence, they're going to attach at one end and release at the other end. So basically activating that cell crawling activates both. It's going to let go in the back and re-grab in the front and pull itself forward.
Okay.
That I don't know. Uh-huh.
Okay.
Wait. The first question's free. Second one's $10. You can Venmo me.
If you send me your Venmo, yes.
Okay.
It's easier through gel though. It goes directly into your account.
Okay.
I was just wondering, like, will people who have cancer and they have a lot of TPA, are they less likely to develop stroke because like they already have TPA in their
That's another good question that I don't have the answer to. I don't know. I wonder. I think I would say no, my guess. Okay, I'm just guessing, but I would say no, because TPA would be like locally released. It wouldn't go systemic like that, but I don't really know. That's a good question.
Okay, thank you.
Sure. Okay, so the almost last thing in this chapter. God, I'm going through material so fast here. I guess because I'm not writing on the board. I've said that before. But anyway, what are the causes of cancer? I think I'll go through the causes of cancer and I'll stop there. So anyway, the very first cause of cancer that was found, again, I'm bringing up my friend Peyton Rouse, was the Rouse sarcoma virus. I just heard a TikTok about it this weekend. God, I love TikTok. There's this woman on TikTok who does the history of cancer, basically, and cell biology, this cellular stuff. But some farmer Peyton Rouse was a molecular biologist, I guess, in New York City around 1910. Some farmer brings him this chicken. He's like, look at this chicken. It's got a big tumor. So Peyton Rouse grabs the tumor, and one of the things they would do back then is filter it, grind it up and filter it. So nothing bigger than nothing as big as a prokaryote could get through. So whatever was left was a virus. Any infectious agent left was a virus. So he took that tumor, ground it up, filtered it, took the filtrate and injected it into other chickens, and they grew tumors. So it was the first cause of cancer that was found. Now, at this point, people believe that cancer was virally caused, which it can be. It's just rare there are very few cancers that are passed by viruses, but it was the first cause of cancer found. So here in present day, we know some virally caused cancers. One is HPV, herpes papillomavirus. There are a lot of different HPVs. A lot of them just cause warts on your skin and stuff. And just a plug for people of your age, reproductive age, make sure you get your HPV vaccine, even if you're a man, especially if you're a man, because you don't know you have it, but you pass it on to your partner. So everyone should get their HPV vaccine. But anyway, HPV is sexually transmitted, and it will infect cells of the cervix, and it can go, latent for a long time, and then stress can make it come out or something. But once it starts to divide, it can be invasive. So it's not commonly invasive, but it's definitely something that needs to be gotten rid of. So only 0.8% actually develop cancer, but I don't want to take that risk myself. So anyway, HPV is one. HPV is interesting how it does this too. So this relates back to what we were talking about with apoptosis and everything, in that it's got two proteins, the E7 and the E6, and a third protein, E6AP. And E7 inhibits RB, which is going to cause the release of E2F and cell division. But as we learned, if you have too much E2F released, you get activation of ARF, alternate reading frame, sequestration of MDM2, and apoptosis. But it puts out this second protein, E6, which is going to block apoptosis. So it binds to P53, brings in E6AP, which is an E3 ubiquitin ligase, and we break down P53. So it needs both of these avenues in order to become unregulated cell growth, because you're activating cell division by blocking a tumor suppressor, and then blocking apoptosis by blocking another tumor suppressor. And Epstein-Barr. So this is another one that I learned through TikTok. I knew about it, but I learned how it was found. So this is an interesting story too. So what's his name? Burkitt. So Dr. Burkitt, maybe you've heard of Burkitt's lymphoma. Dr. Burkitt was a surgeon in Africa, and he saw that there was this common lymphoma in a lot of children in Africa. And so he was studying it, and Burkitt's lymphoma becomes a different type of lymphoma. But at the same time, these two researchers, Epstein and Barr, found this virus that was associated with it. Epstein is a man, Barr is a woman. Of course, we're out of Women's History Month, but I think we still should celebrate the accomplishments of women. So Epstein and Barr found this virus, and it puts out these two proteins, LMP1, it's activating BCL2 production for anti-apoptosis, and it's also activating cell division. And LMP2A is also activating cell division through this the RAS pathway, which RAS can also activate PI3 kinase, and then AKT. And now here we've got the JAK-STAT pathway activated. So these proteins then are activating those pro-growth regulators that are causing the cell to divide. So anyway, bottom line, long story short, viruses can cause cancer. Not that common necessarily, but it is a possibility. Thank God it's not common, right? Viruses. Then we got to get vaccines, and then it becomes political. Okay, so proto-oncogenes. So I talked about this a bit earlier today. Proto-oncogenes are basically any gene that will produce a protein that causes cell division. So this is everything from the growth factor all the way through to the transcription factor. So there are some oncogenes. VCIS is one, which is just basically PDGF. So the cell starts putting out, it gets VSIFs means it comes from a virus. So there's a virus that causes the production of PDGF, then you get autocoid stimulation. Receptors, ERB, which is your EGF receptor, TRAC, we'll talk about TRAC shortly, nerve growth factor, epidermal growth factor 2, thrombopoietin, which, as you can probably assume, has something to do with blood cell division. So these are receptors. Then you talk about G proteins, which are the next step in a lot of growth factor pathways, KRAS, HRAS, and NRAS. These are all mutations of the RAS protein. And then your kinases, BRAF, BSARC, SARC itself, JAK-STAT pathway here. We'll talk about this, BCR-ABL. That's a really important one as well. And then transcription factors. You remember when we were talking about MYC, how MYC alone can cause the cell to become, start to divide and become out of control. So there are a lot of different forms of MYC, mutated forms of MYC that can cause the cell to go into division. Jun and Phos, we talked about being activated by MAP kinase. Cyclins and CDKs, right? They're out of control. The cell keeps dividing. So basically everything down the row from the hormone to the IEGs to the DEGs, if they're out of control, then they're going to cause cancer. Now, how do they get out of control? DNA mutations. I mean, typically we think of cancer as a disease of DNA mutation. So I'm hesitating here because I think there are new theories about it that contradict this, but at this point we're still kind of under the philosophy that it's DNA mutations that cause it. And how does that happen? A lot of different ways DNA can become mutated to cause a proto-oncogene to become an oncogene. So keep that in mind. Mainly what we're talking about here is activating proto-oncogenes. Usually when we think about mutations, at least when I think about mutations, we think about point mutations. Point mutation is when you change one base pair. And when it comes to point mutations, you know, most of them are going to be silent, right? They'll be either in non-coding regions of the DNA or in introns of the genes. But in general, they're not going to cause any changes in cells. When they do cause a change in amino acid, most of the time it's going to inactivate that protein, just shut it off. And that's not going to cause cancer. But there's the random rare chance that a point mutation can cause a change in an amino acid that activates the protein. So it's rare, but it does happen quite a bit. You know, rare, but there are 10 billion events a day that happen in your body. So no matter how rare it is, it can still happen. And one example is RAS. RAS is classic for this. If you mutate out glycine 12, glycine 13, or I think this is glutamine 61, then it no longer interacts with GEF. So once it gets activated it no longer interacts with GAP. So once it gets activated by GEF, it stays activated. So that's an example of a point mutation then that can cause cells to become active. Another example is with receptors, right? Normally growth factor receptors don't interact with each other unless the growth factor is around, and then they dimerize and activate. There are mutations that take the receptor off, so you just have the transmembrane and the kinase, and that somehow causes them to dimerize in the absence of growth factor, and then they become constitutively active. So those are examples of point mutations causing activation. Another thing that can cause activation is gene amplification. So as you know, in S phase, when the genome is being replicated, all kinds of crazy stuff happens. I mean, you like to think it's going to be fidelitas. Is that a word? I don't know. Fidelitas. You like to think it's going to be fidelitas, but you know, the template wraps around and it gets it gets copied twice instead of once, and you end up with multiple copies of a gene. When you look at the Human Genome Project, they found all kinds of genes that are just replicated over and over again. So anyway, this can be a bad thing if it's a proto-oncogene, because sometimes just making more of that protein is going to make it converted from a proto-oncogene into an oncogene. An example of that are receptors. If you get an awful lot of receptor, you overproduce the receptor, it no longer needs growth factor to activate. They start bunching together and activating themselves in the absence of growth factor. Another way to get overexpression is translocation. So now we're talking about big changes in genomes. So thinking back to when we talked about dysplastic cells, I said that the nuclei look weird. So as cells get more and more sick, then, you know, a lot of the controls over DNA replication get lost. And so, you know, when normally the cell would stop and fix its DNA, it just keeps going and crazy shit happens, crazy stuff happens, sorry. So one thing that can happen is a chunk of a chromosome could go into a different chromosome. Here's an example of that. So here, not an example, but a nice figure on it. Here's chromosome A, chromosome B. This one chunks here, this one breaks here, and they switch over. Now this can be bad because, you know, going back to what we said with eukaryotin and heterokeratin, eukaryotin is all condensed up like this, so it can't be activated. And genes that should be silenced are usually contained in that eukaryotin. And you put something that should be condensed into an area that is open, and now suddenly you're activating that protein. Let me show you what I mean in the next figure. So this is Burkitt's lymphoma. I was talking about Burkitt's earlier. This was his discovery there in Africa. So MYC, remember our old friend MYC. All you need is MYC and the cell is going to divide. So it's on chromosome 8 in an area that's going to be condensed up and controlled until it's needed. On the other hand, you've got on chromosome 14, you've got a region where immunoglobulin heavy chains are made. So if it's a white blood cell, that's going to be an active area, right? It's making a lot of antibodies, so it's got this area all this is condensed and this is opened up. When these two swap, so now you've got a chunk of 8 on 14, you're putting MYC into an active region. It starts to get transcribed and you get a lot of MYC, and that's where you get the lymphoma from. So that is an example of translocation activating a protein, mainly by putting it into an active region of the chromosome, and then it gets overexpressed. Here's another one of chromosomal translocation, but this is even crazier. My stepmother-in-law has this. It's called the Philadelphia chromosome. It causes a chronic myelogenous leukemia, which is not that bad, but it's bad. But it's definitely going to advance to acute myelogic leukemia, which is deadly. So chronic makes you tired, and you'll be tired for about 5 years until it goes acute, and then you die in 2 months or something. So this is a really nasty leukemia. And it happens because of a translocation between chromosome 9 and chromosome 22. And then it makes a new chromosome called the Philadelphia chromosome. And here's why that's bad. So BCR is a kinase, okay? It's got SH2 binding domains, Rho GEF over here to activate Rho. Most importantly, it's got a serine threonine kinase. So you can think of this as like a RAF or a MEK or a MAP kinase. And it's normally regulated. But it'll break off there from that chromosome 9 and connect to this end of the ABL protein. ABL is another kind of scaffolding type protein. You can see SH2, SH3, tyrosine kinase here and SH1. So another kind of crazy protein. You splice these two together, and now the serine kinase is no longer regulated. And so it just causes activation of signal transduction and cell division. So that's your classic chromosomal translocation, causing the production of a brand new protein, which is causing the cell to be out of control. So that's another way. Oh, here, I have one more figure here. So here is your new protein, and there are a whole bunch of really awesome drugs here that keep people alive. My mother-in-law was on this one for a while till she got side effects. And now she's on some of these. But, you know, drugs are drugs. She's having some cardiac side effects now. So it's uncomfortable. Okay. Oops, too far. So similar kind of a thing can happen, but just in one chromosome. So we're not jumping between two chromosomes, but just in one chromosome, you could get a chunk of DNA inserted, a chunk of DNA taken out, or chunk of DNA taken out, flipped around, and put back in. Oh. So here's just a deletion, big chunk deleted out. Here's an inversion, flipped around, going backwards. So you can imagine taking a chunk out. It could, again, put a proto-oncogene from a quiet region into an active region, causing overexpression. Or, you know, you can imagine a lot of different ways. Just losing a big chunk of the chromosome could change the regulation of that chromosome and the genes on it. There's a really interesting example of what this can do. Imagine you've got one protein here, so you're right in the middle of the gene itself, right in the middle of the coding region of the gene here. Different gene over here, right in the middle of the coding region. You flip this around, you make two whole new proteins, and that's what happens with this. What's it called? End track. So one end of it is the NGF receptor, right, which is a membrane receptor, and the other side is tropomyosin. Remember tropomyosin, chapter 3, is a coiled coil protein that covers the myosin binding sites on actin. And so it's going to spontaneously coil. And here's your NGF receptor. Normally, to get them to dimerize, you need FGF. So what happens is this breaks here and here, flips around, and now you've got the TRAC tyrosine kinase and a coiled coil. So they automatically bind to each other and automatically activate. So you've got a constitutively active growth factor receptor because of that. I just love that mechanism because it's so interesting the way that happens. It puts two proteins together that then let me turn off then cause it to be active. Okay, and now our last thing is retroviral insertion. So retroviruses, I do have a figure here. Retroviruses, basically like our HIV, are going to fuse with the cell and go into it, uncoat, and then they're RNA viruses. So one of the first proteins they make is RNA reverse transcriptase. They make DNA. That DNA then inserts in the genome of the host, and then it continues to make more RNA, more proteins, and more virus. So that's your life cycle of the retrovirus. This integration is the part we're concerned about here. So the virus will insert itself somewhere randomly into the DNA. And this is going to be an active piece of DNA. This virus is very active. So what can happen is I don't have a figure. So what can happen is that viral DNA can insert right next to a gene that's really important to control, like MYC. So MYC is in a quiet area. You put a retrovirus next to it, and now you got MYC. So retroviral insertion can also activate cell division. interesting story. When I was finishing grad school in 91, one of the new things people were thinking about was retroviral mediated gene transfer. So you know what the mutation is that's causing a certain genetic disease like cystic fibrosis. You put that gene into a retrovirus, you infect cells, you put the correct gene in. Great idea, works great. Problem is, there's no way to direct where that retrovirus is going to insert into the genome. So you have, knock-on effects in terms of cancer being caused. So it was a great idea, but it never really panned out. Now we've got CRISPR, which is site-directed. So it's going to go right to the place you want it to go. No more problems with that. So anyway, that's about all I want to talk about today. I feel like I talked my head off today. We got a lot done. Let me stop my share and we can talk about what we went through, and then we can review it again next time. Any questions about what we did? Nobody's got any questions? Okay, so why don't we call it a day? I'll post this up later today. And I'll see you again on Wednesday. Again, it's going to be online Wednesday. I've got course advisement for my undergrads this week. So I'm going to be going in Monday through at least Thursday, if not Friday. So Wednesday will also be online. We all set? We all good? I'm going to say goodbye.
Thank you.
You're welcome.